I'm excited to introduce Dr. Paul August. Is he in the room? Paul, wonderful, fantastic. So Dr. August is the Chief Scientific Officer at ReviR Therapeutics, and he's previously held leadership positions at Agios, Icagen, and Sanofi. He also is a founding member of the CMT Research Foundation's Board of Directors, and as well as being a valued member of our Scientific Advisory Board. Please join me in welcoming Dr. August and the panelists to the stage.
Great, thank you, Keith. Well, let's bring our panelists up. I am so excited to be hosting this, moderating this panel discussion today. You know, as you heard from, the board members and Keith and Cleary, we have been working towards raising funding to support the development of therapeutics. And here on the stage, we have a number of sort of executives, senior biotechnology company executives who are developing therapies for CMT. And for some of these, we've been involved in sort of helping to support their efforts through your generous donations. And today, what I'm going to do is we're going to split this session into three parts. In the first session, what we'll do is we'll have each company introduce themselves, and you'll hear a brief overview from each one of our guests here about their company and their efforts in working on CMT. In the second session, I'll ask some of the companies some very specific questions about some of their efforts, maybe some that you'd be interested in asking yourself. But then after that, we will kind of open up the microphones for both the live and the virtual audience to ask your questions again. And if you're online right now, please feel free and send in the questions, and we'll start with those. Rob will gladly deliver those to us, and we'll start with those when we move to that session. So we're fortunate enough today to have Artie Suckow from DTX Pharma. He's the CEO working on antisense type approaches to treat CMT1A. We have Scott Harper, who is the Chief Scientific Advisor at Armatus Bio. We're also fortunate to have, and I had a great conversation with earlier, Thomas Holm Pedersen, CEO of NMD Pharma, and he came all the way from Denmark. So thank you for traveling here today. And then we have Sylvain from Augustine, which is from Belgium, also in the same area. And we're fortunate to have you. Of course, no relation to me, Paul August. So, but it's a great name. I do love that name. And then we are finally lucky to have Chris Lorson from Shift Pharmaceuticals. And Chris, this company is working on nucleic acid therapies. So let's start working our way across, and we'll talk to the panelists. And why don't we start here with you, Chris, and then we'll work our way across and introduce yourself, a little bit about your company, and maybe kind of some of the approaches that you're taking to CMT and what some of the subtypes that you are working on.
Great. Thanks so much, Paul. My name's Chris Lorson. I'm with Shift Pharmaceuticals, and Shift has been around for about 5 years. The co-founder and current CEO, Steve O'Connor, is in the back. And this has been something where Shift has been focused on developing antisense oligonucleotides, or ASOs, that modulate gene expression. So that's a lot of words, but what I really mean is that ASOs are synthetic nucleic acids. They're short, typically 18 to 22 nucleotides. And in our case, we're trying to target pre-mRNA. And pre-mRNA is that stuff that's made from the DNA. It's more or less a duplicate copy of DNA, and it's the precursor to that book that you will use to make a protein. So we want to kind of use antisense oligonucleotides to redirect the reading of the chapters in that book. And the way that we do that is We have a drug for SMA and spinal muscular atrophy. We want to increase the inclusion of a particular exon or basically reading a particular chapter more frequently. Or in the case of CMT, we want to skip a particular chapter, and it's all targeting specific regulatory regions. It's very specific. It targets just that gene, that genetic region. And that's kind of the approach that we're trying to do. Some of the benefits to antisense oligonucleotides, or at least in this case, they're remarkably stable. The backbone for these are remarkably inert. So they've been in Duchenne's muscular dystrophy for about 25 years. So in terms of safety, tox, those kinds of things, the backbone that we use is remarkably stable and remarkably low in terms of toxicity. I think it's really great. Shift has received funding from CMT Research Foundation. It's the reason why we got into the field. We were effectively recruited into the area. And I think that that's one of the things that you really see is that reaching out and bringing people in from other fields can be a very powerful way to do it. I was in the SMA space before, and that's the way the SMA field did it too. It was a relatively small community, and then really focused, organized research foundations brought in people from outside the field and brought in their expertise. So it's really been great. I've not been to a CMT conference before, and I feel like I've been trying to kind of like take a drink from the fire hose. So it's really great to really learn so much at this, and I really appreciate the opportunity to be here.
Well, thanks so much for being here, and thanks for that. Let's hand it over to Sylvain. Can you tell us a little bit about your company?
Yeah. Hi, I'm Sylvain Celanire. I'm CEO of Augustine Therapeutics. We are based in Belgium. So Augustine is, in fact, a spin-off company of the VIB-K11 Institute, so an academic labs, which is a hub in a lot of CNS disorders. The company has been incepted in 2020, so nearly more than a bit 2 years. I've been joining the company from my past years in I would say, drug discovery and development in CNS and PNS field as well. So we are developing small molecules, so we would like to develop an oral pill that is targeting HDAC6 inhibitors. HDAC6 has been a key, I would say, enzyme that are regulating a lot of, we say, dysfunction and primarily axonal dysfunction. Should it be primarily in like CMT1 or we say secondary degeneration as well? So we are at the preclinical stage. We have a lead candidate that's moving forward, and the goal is really to target all type of CMT. It says we have the luxury that we have already data that support CMT1A, CMT2A, CMT2D, CMT2F data that are supporting with using patient cell line, but also animal model for moving forward this therapy for all type of CMT. And the goal for us is to embark in another story with maybe CMT1X. So that's where we are now.
Great. Thank you, Sylvain. Can you hand them? Oh, you've already got your microphone there, Thomas, but maybe you tell us a little bit about your company.
Yeah, so thank you very much. So first of all, thank you very much for inviting us. I think it's been a fantastic meeting so far. I've definitely learned a lot, so thanks a lot for that. My name is Thomas Pedersen. I'm CEO. I'm also founder of the company called NMD Pharma. We're a Danish-based company, but we do actually have an office as well here in Boston. We're at the clinical stage. We are a company that's focused entirely on neuromuscular diseases, and our main program focuses on developing a small molecule inhibitor of an ion channel called CLC1. It is a chloride ion channel that is exclusively expressed in skeletal muscle. Now, what you gain by inhibiting this particular ion channel is that you basically enhance the communication between the motor nerve and the muscle function, or the muscle. So thereby you bring back muscle function, bring back muscle strength, hopefully being able to basically improve mobility, strength, and also reduce fatigability or reduce fatigue. So we're at the clinical stage. We have just completed a phase 2 in myasthenia gravis, and this data is coming pretty soon. Now, this mechanism we're developing is not necessarily linked to any particular neuromuscular disease. We can actually use that, that's what we hope, for a range of different neuromuscular diseases, and therefore also for a range of CMT subtypes. However, we were not actually that confident that we could go into CMT as an indication per se, because we did not know so much about the neuromuscular transmission. in CMT. So what we decided to do was to run what we have learned about earlier today, and clinical observational study. So we did that. It's a study that we called it the established study. It was a study where we had two centers, one in Ohio, and we also had one in Denmark. And what we did there, we basically used established biomarkers of electrophysiological signals, specifically something called single fiber EMG, to determine to what level CMT patients have neuromuscular transmission deficit. And I can say, while we haven't published that yet, that we do see that there is quite profound neuromuscular transmission deficit in the patients we have been working with. Now, what that does is it actually triggers us to say, well, this looks like that the new mechanism we are developing might be applicable for CMT. So we are moving forward. We have our phase 2 in myasthenia, but we now also want to expand into CMT. So we're a little bit new here in CMT, but it's definitely something that we really hope we can contribute to as well that field. So thanks a lot for having me.
Well, it's great to have you in the CMT field. You know, it sounds like it has a lot of promise. Artie, please tell us about DTX Pharma.
Yes, so I'm Artie, and like Chris, I was recruited by Susan and Paul to come to the CMT1A field at a conference several years back. At DTX, we also work on oligonucleotide therapeutics. We work on a different flavor than Chris. We work on something called RNA interference, or siRNAs for short. And the fundamental problem with that class of therapeutics is that cells don't take them up, and they're rapidly cleared by the kidney. And so we have a technology we call Falcon. It's named after my high school mascot. Kind of forced it, but it's called fatty acid ligand conjugated oligonucleotides. And all that really means is we attach fatty acids directly to the siRNA to overcome the two obstacles. So what the fatty acid does, it enables these drugs to get into cells. So you can use these receptors or transporters to kind of translocate them into the cell so they can do their thing. In our case, repress the expression of PMP22 in CMT1A. And the other thing that it does is, since they're rapidly cleared by the kidney, you can actually, when you inject these fatty acid conjugated siRNAs, they hop a ride on a protein in the blood called albumin, and it's kind of an Uber driver. It prevents clearance by the kidney. It drives you around the circulation for a lot more times and drops you off, in this case, in the peripheral nervous system. And, many of you have probably heard me speak before, but, from pilot study all the way to today, the studies were initially funded by the CMTRF. We raised quite a bit of money to take this molecule into the clinic, and hopefully in less than 12 months, we'll take the molecule into clinical development in the next, probably less than 12 months. So.
That's so exciting, Artie, to move from where we were a few years ago in your presentation last year to being in the clinic next year. We're so excited. Chris, or I'm sorry,
Scott.
Scott.
Yeah, my name is Scott Harper. I am here as the Chief Scientific Advisor of Armatus Bio, who's one of the sponsors of the meeting. But my life began in academic science, and it still remains there. So I'm still relatively new to the CMT field as well, although I've been flirting with it for about 15 years. And so there's a backstory to that. I don't know if I have enough time to tell you about, but I can give you a little flavor about how we entered CMT, but also what we're trying to do. So maybe I'll start there. So you heard from Chris and Artie that they're working on using nucleic acids, essentially DNA or RNA or modified versions of that, to try to deliver those to Schwann cells and reduce expression of PMP22, which is this, the gene that underlies CMT1A. So it was already mentioned that delivery is a difficult thing. It's actually one of the major bottlenecks. We need to be able to, if we're going to be using these nucleic acids for therapies to knock down genes, we need to get those to the right cells. And one of the challenges here in anything that we're trying to do is that, we have trillions of cells and they all have these membranes around them. And the membranes are designed to try to keep out nucleic acids. They're not, we don't, our bodies are trying to keep our DNA in and prevent other DNA from getting it. So that becomes the challenge. How do we get these nucleic acid drugs across that cell membrane with Schwann cells so they can actually do their job? And so, you heard of this Falcon technology. Chris is using a technology with Shift that I'm not sure of the mechanism how it gets in, but it's an uncharged molecule and it must cross the membrane that way. We're taking a different approach. So what we're doing is gene therapy. And what that means is we are using natural viruses. We're trying to modify these viruses to try to deliver therapeutic genes to Schwann cells. And so the advantage, there's advantages and disadvantages to this approach. The advantages are, we're able to use, so these guys are being very inventive in figuring out ways to deliver things to cells. We're using natural processes that viruses have figured out over 200 million years how to do. So we use in particular a virus called adeno-associated virus. It's abbreviated as AAV. These are viruses. There's actually two products approved using AAV vectors. Now one's a treatment for blindness. That was the first gene therapy approved in the United States. And then it's pretty miraculous, actually. It's literally curing blindness in kids using this virus to deliver genes to the eye. And then there's spinal muscular atrophy. There's a product called Zolgensma that was actually invented at Nationwide Children's Hospital, where my academic position is. So, okay, so these viruses, over hundreds of millions of years have acquired mechanisms to deliver their DNA or RNA into in different cell types. So the AAV viruses we're using, they like to deliver genes to muscle and nerve and eye and other things. So what we're trying to do then is to package a therapeutic DNA construct that is designed to knock down PMP22 and reduce its levels. We're packaging that inside a virus, these viruses that have a natural ability to infect Schwann cells. And then we're growing that virus up in the lab and delivering it to, in our case, animal models right now. And they naturally find their way to Schwann cells. And then they can deliver the therapeutic product there in Schwann cells. So the major advantage to that is we're using this natural process to get things into cells. And if we did this right in a perfect world, and I'm not saying that this is how it's going to end up being, but the dream of gene therapy is to have a one-time treatment. So we do one delivery, that virus delivers the payload to cells. And if that cell is not dividing, it should last essentially forever. The longest we know so far in the clinic is 10 years, how the longest these have survived. So, yeah, that's what we're trying to do. And I'm happy to be in the field. I've already talked long enough, but I just want to mention that, the work that we're doing at Armatus, we're working on two different gene therapy projects for CMT. One is targeting CMT2D, so GARS, and another is targeting CMT1A. And I'm just riding on the backs of two really amazing people who are already in the field. Rob Burgess, who's a GARS to be able to collaborate with both of those labs, and happy to be here now talking to you all. I guess I'll stop there.
Okay, thanks so much, Scott.
Sure.
I really appreciate that. It was a nice in-depth overview of a little bit different modality than we have here on the stage, but nevertheless, a really important one and critical one for rare genetic diseases. So what I'm going to do now is I'm going to kind of give each one of you maybe a question and, answer that. And if any of the other panelists, feel like you want to contribute to that, please raise your hand. We'll pass the microphone to you and contribute. So I'll start with you, Chris. So Chris, you study a variety of different genetic diseases. You've been involved with the development of therapeutics for SMA in the past half decade. Where do you see the progress in CMT relative to some of the other genetic diseases that actually have treatments today?
Not that the question was leaked to me, but I happen to have a few notes. But I do think that on multiple levels, there's a lot of crosstalk. And I think that for rare diseases, it's really important that you really leverage the strengths of the community. And so while the genetic targets are clearly different, within CMT, SMA, ALS, it's really important to learn as much as you can. And so I think that a lot can be learned, and that really identifying good clinical targets is essential, and that developing good clinical trials that ask the right questions is critical. The patient population isn't enormous. We're not talking about cancer, a common cancer. These patients are very, very precious, and asking the right question is essential. You heard a lot about that today, and I think, these are going to be beautifully planned, but I think in SMA, that was one of the things that really allowed drugs to be approved. And it was trial design and thoughtful trial design beforehand. You want to be able to keep the good and also remove the bad. So setting up trials to ask those questions. In SMA, spinal muscular atrophy, there are actually 3 approved drugs. There's an antisense oligonucleotide, a small molecule, and a gene replacement. So an AAV9 SMN replacement. So a rare disease with multiple drugs approved is pretty remarkable. I think the next thing that you're seeing also applies to CMT is that there's also a wave of drugs that will be non-SMN targeting. SMN is the gene that's missing in SMA. Gene modifiers is going to be a big one, or gene modifying pathways will be really important. I think that if you just kind of look at the CMT community, there's a number of things that I think are really important. The animal models are in place. The patient community is active and ready. You can kind of sense it, that there's an urgency, and I think that's really important. And then also having organizations like the CMT-RF that drive this kind of interaction and set up a community, that it's not just academics, it's biopharma, it's government relations, and CMT-RF serving as the grease to really make things happen, I think is really critical. And you can tell that this is something that's really important to this organization.
Great. Thanks a lot, Chris. Does anyone want to add anything to that? Okay, great. Sylvain, your company studies HDAC6 inhibitors, and HDAC inhibitors are also being studied in a variety of other diseases. How are the learnings in studying these drugs and other diseases informing, your work for CMT?
Yeah, so indeed, the first asset of the company was HDAC6 inhibitor. It has been actually, everything starts with a patient, and Augustine is the first patient of Professor Suckow in Paris. So we start with a name, with a patient. And the second thing that, although we have some scientists that are patient, and actually we have Rob prior that has been part of the story of Augustine, working with Ludo van den Bos and Vincent Timmerman, initiative of the Augustine, I would say, story in identifying HDAC6 inhibitor, I would say 10 years ago, when they discovered the role of HDAC6 in CMT2. So it has been the starting of the company where we moved to now to explore the role with the application of HDAC6 in many subtypes. And so we have been learning from the CMT2 field with Vincent, Ludo van den Bos, which is the scientific founder of the company. What we learn is that there's an axonal, we'd say, degeneration in many diseases, and CMT2 is one of them. We learned that there are many subtypes with axonal degeneration, but it's a common denominator of also in CMT1A. And so we bring CMT2, I would say, application of our HDAC6 inhibitor, the new generation of compound that has never been studied so far. So we are, I think, developing the third generation actually for the past generation of compounds for the past 10, 15 years. And moving from CMT is also how we can apply that to other indication. We have now the proof that we can also treat chemotherapy-induced peripheral neuropathy, which is also a link with CMT for some patients. And we move forward with now a new generation of compound for ALS. So what we know from the CMT field and the data that has been generating with a compound, we can apply that to other indications. So CIPN is the second line of our pipeline, and ALS will be the third line of the pipeline as well. And we have the luxury to work really closely with Ludo van den Bos and Philippe Van Damme at the VIBK Leuven and the hospital in Leuven. They have a center for CMT. It's also a center for ALS. And so we have the chance to get some patient cell line. We can, in fact, look for iPSC cells and effect in patient cell line to see all these compounds can really benefit and make a change for curing CMT, but also other diseases. So it's been a great, I would say, story since we started 2 years ago. Now we are in preclinical development, and our compound are actually demonstrating a lot of benefit in any of these subtypes for now. And there's a really hope that we can change something in the near future.
Oh, that's fantastic. Do you see that, sort of this advancement in multiple indications helps to develop the biomarkers that you can use for the development of your therapeutic?
Correct. We have been learning that a lot of yesterday and today as well. One of the key biomarkers I've been using in many clinical trials, which is NFL, so neurofilament light chain.
Mm-hmm.
So we have been doing translational science from the preclinical to the clinical, but now we have back translational, what has been learned from using NFL and other biomarker from the clinical stage, how we can have that in our preclinical model to strengthen the case for a compound to demonstrate a benefit. So NFL is one of the key biomarker we are using. We have the chance that working with HDAC6, we have another biomarker, which is tubulin. Tubulin is one of the main substrate of HDAC6 that, in fact, stabilize macrotubules of the peripheral nerve. So we can do a blood sampling maybe in animals, but that will be done also in human. So we just make a blood test measuring tubulin acetylation, which is a target engagement assay to measure that the compound is on board and make an effect. So biomarker has been the driving force of going forward.
Oh, that's fantastic to hear. Thomas, you and I spoke earlier about your company, and you told me about your efforts kind of developing drugs that cause the skeletal muscle to react more strongly to signals that are coming from the spinal cord. How might that be beneficial in a disease like CMT that affects the nerves?
Yeah, so fundamentally, you could argue that CMT, of course, is a motor neuron disease, but I think it's very, very important that we think of this as for what it is. It is basically a very long cell that is relying on support from basically from the soma, from the cell, and then a very long path of transport down the axon down to the terminal, and the terminal has to basically be the point of contact between the muscle and the nerve. Now, that means we have a basically, you could say, a crucial part of this whole motor system lying at the very distal part of the motor nerve. So we believe that the nerve terminal is going to be affected in most diseases where there is a motor neuron challenge. That be either axonal degeneration, demyelinations, axonal transport issues, many different things. We believe that it will result ultimately at the neuromuscular junction. Now, we know also, of course, that CMT is a denervating disease where there is constant efforts to re-innovate. So you have this situation of denervation, re-innovation that is not at sort of in a normal situation. So you have both ongoing denervation, but also ongoing re-innovation. All of that actually leads to unstable motor neurons, contact between the muscle fiber and the motor neurons, so at the neuromuscular junction. You basically get unstable neuromuscular transmission. That is something that we know very well. If you have that kind of dysfunction at the point of contact between the motor neuron and the muscle fiber, that can lead to weakness, it can lead to fatigue. So what our approach is doing is it basically brings back that function, it brings back functional boosting, you could say, at the neuromuscular transmission, making it possible for the motor neurons to again activate the muscles. So that brings back muscle strength. It brings back hopefully endurance. And it is important to say, I think, that the target we're addressing, again, this CLC1 chloride ion channel, is something that you only find in skeletal muscle. So it's very specific. So to your question, what we hope it does, it brings back the ability to activate the muscle, bring back strength, using a very selective target only expressed in skeletal muscle.
Oh, that's great. So it sounds almost like there's a reinforcing, of working on the muscle, reinforcing the neuron and enhancing that whole network.
I think there you're talking about something which I think is super, super interesting. The whole concept of bringing back muscle function long-term, how is that actually going to affect also working back retrograde on the motor neurons per se? Because even just the ability to sustain functional, being more functional, how is that actually going to improve the whole motor system? I think it's well known that if you have a loss of contact between nerve and muscle, you have very rapid atrophy. So sustaining functional
Right.
probably could lead to better integrity as well.
Yeah, absolutely. That's fantastic. Scott, we're going to shift to you. You mentioned to us about, some of the work that you've done in gene therapy, and you've created genetic medicines for several genetic diseases. You worked on Duchenne muscular dystrophy, vasculoscapulohumeral dystrophy, and now Charcot-Marie Tooth. And you're also very active in sort of leadership positions in the professional societies, such as the American Society for Gene and Cell Therapy. Can you tell us about one or two recent developments in gene therapy science that are having an impact on the way biotech companies are approaching CMT?
Sure. Yeah. So I mentioned earlier in my opening comments, I mentioned these viruses that we're using for gene therapy. So, I also mentioned that I flirted with the CMT field about 15 years ago. The short version of that is that I was 15 years ago, I was starting my lab as a new independent investigator and was looking at the technologies we were developing. I thought we could apply to a number of different diseases, dominant diseases. One of those was CMT1A, and tried to get funding for that program back then. And one of the major problems was this was still sort of in the early days of these AAV viruses being developed. And we didn't have a good AAV virus that could, the word is transduce, but it essentially means infect Schwann cells. So, that was actually a major barrier. If we've got this genetic medicine that we think works, but if we can't get it to the right cells, it's not going to work. It's not going to be functional. So there have been a number of advances in, number one, identifying new viruses that are just out there in nature that can target Schwann cells, number one. And then also it's actually a really hot area of trying to do designing of these new viruses to engineer new ones that don't exist in nature, that might have better properties for binding Schwann cells and delivering payload to that. So I think those kind of advances in that vector delivery system, I mentioned delivery is the bottleneck for a lot of these gene therapies. That's a major push in the field, and it's really exciting. Now we're using what's called AAV serotype 9 to deliver genes to Schwann cells, but we and others are looking for better and trying to engineer better systems for improved delivery.
And that's something that biotech companies or pharma companies see as kind of a major hurdle that
it's very hot. Yes.
Okay. Great. Artie, your company's approach uses a form of RNA to reduce the amount of PMP22 for CMT1A. And your secret sauce is that you combine RNA with fatty acids. Why do you use fatty acids, and how really does this improve treatment?
So kind of alluded to why we use fatty acids in my intro. I think the short answer, or maybe it's a long answer, on how we got to fatty acids is earlier in my career, I used to work on small molecules that target fatty acid receptors for a totally different disease, diabetes. And I thought there was probably ways you could hijack these receptors to bring these molecules into cells. And so you all might not be familiar with this, but in the liver, there's a sort of similar technology called GalNAc, which is a sugar you couple to the siRNA. And there's, $100 billion worth of deals and approved products using this sugar to deliver siRNA. So, I thought, like, sugar is very simple. Let's try to use fatty acids. And of course, the second piece of the puzzle when I was talking about the Uber is that there was 4 multi-billion-dollar drugs in the diabetes space that leverage fatty acids to do what I said before, take drugs that would otherwise be cleared by the kidney in a few minutes and get them to last as much as a week in the circulation. When you come back to the problem with siRNAs, even ASOs to some degree, is they just are filtered through the kidney as quickly as you can put them in the body. And so your Schwann cells of your peripheral nervous system never, ever see these drugs without some help. So I guess the elevator pitch version is fatty acids, every cell in your body takes them up, So we could hijack those mechanisms. We can keep them around longer in the circulation. And that's ultimately how we think they work.
And you use and explore a variety of different fatty acids in your work, and you have to discover which fatty acid will work for the disease that you're working on, right? Is that part of
Yeah,
your...
so I think, well, when we first met Susan and Paul, we had kind of one, we call it a motif, one sort of chaperone that we attached to the siRNA. But what we've learned since then is sort of the fatty acids that make up that chaperone, if you will, for lack of a better word. Actually, the best ones for, say, the brain are different than the best ones for the muscle, which are different than the best ones from the peripheral nervous system. So there's, you know, lots of optimization that kind of goes into enabling a new therapeutic area or a new tissue, if you will.
Really kind of like programming the siRNA to go where you want it. It's a really interesting technology. Sylvain, I'm going to switch a little bit in terms of gears here, you know, and talk about sort of the pipeline in CMT. As you look across the landscape of the therapeutic pipelines in the CMT 1A space, how do you feel about the competition that you're seeing to develop therapeutics? Are there sufficient therapeutics that are in development? Is there room for more therapeutics to come? And is there sufficient capital to develop those therapies for CMT 1A? I know that's a lot of questions, but, you know, have that.
I'm sure I will be helped by the others.
And if anybody
That's
else
fine.
wants to jump in, and please feel free.
So yeah, the pipeline of therapies is there. So small molecules, gene therapy, cell therapy. You have a beautiful pipeline on the CMT Research Foundation that help, I think, patients, but also investigators and researchers and pharma companies to see where are the competition, which target are the most popular, where are the hurdles for some of them as well. So there's definitely room for many other, I would say, mechanisms of action to treat this CMT, whatever the subtype, not only CMT1A. This is what we can see. There's nearly, I think, 15 projects that are targeting and pharma companies developing drugs, should it be gene therapy, cell therapy, or small molecules targeting CMT1A. There's many also target for CMT2 as well, less for CMT1X and CMT4. But definitely, there's a lot of, I would say, enthusiasm in new mechanisms of action. The downstream pathways are always different. Should it be at the neuromuscular junction, as Thomas mentioned? Should it be at the axonal transport defect, mitochondrial defect? There's many pathways and way to address the CMT pathology. And I think there's many more, I would say, target class that are within the pipeline of big pharma companies and biotech companies and academic research to bring forward new therapy and new hope for treating these diseases. I think what we learn from what are the player in the field, there's not so much big pharma in that area. That's true. I think biotech companies are taking most of the challenge to move forward this therapy to the commercialization and to the marketing authorization, but they need a regulatory path to that. And we know Pharnext is one of the most advanced today. They are taking all the orders of the regulatory approval and pass and clearing everything. So I think they are taking a lot of time to move forward this triple combo drug to the clinic and as well to the market. If there's one success, that will be at least a success for everybody because the path is there. Mm-hmm. I think we need competition. We need that to bring forward many options for patients. And the capital, the funding that we need for clinical trial, but also for drug discovery is large. That's true. If you look at CNS disorders, neuropsychiatric disorders, you could have only maybe 2-3 weeks, I would say, or let's say months of treatment. For CMT, you need years of treatment to be sure that you have I would say, a minimum effect to be seen. So running clinical trials is really expensive for biotech company alone. And so as we in other, I would say, indication, you need to have, I think, plenty of partners that can do that all together. If we compare to Alzheimer's disease field, now big pharma companies are merging together just not to be alone. There will be Biogen, Eisai, whatever company has been merging effort for a single treatment for antibodies and other therapies to move forward. For CMT, it should be the same. I think we need the help of big pharma to move forward with biotech together and facilitate the clinical development of such drug. There's no, we say, a lot of expertise, I think, in CMT field precisely in big pharmas now. That's why they are asking many questions of the way forward for biotech to be there. But definitely a lot of capital for this rare disease is important. And there's many associations that are critical. CMT Foundation is making, I think, a first in that direction. But we need more, I think, definitely. And there's plenty of hope for that.
Great. Thank you very much. Does anyone else want to say anything about the capital markets or this question?
Well, I can say a couple of things here. So I would say, first of all, it's true that that we don't see that many big pharma players in the CMT field right now, but I don't think it reflects lack of interest, actually, to be honest. I would say there is definitely a big interest for CMT, and I think that's good to know, because it means eventually, things like what we guys are trying to develop would at one point probably be, or could perhaps be taken over by a bigger party, and there would be interested parties to take over something like that. And ultimately that may be the way that is going to get to the market in the end. So there are bigger players out there that have the interest for it. I would say in terms of capital, as a biotech, it's always capital that's a limiter. It's always a limiting factor is to drive things forward. I mean, that is the way it is. Hiring people, running clinical trials, doing all of the CMC activities, whatever it may be, it is just very expensive. And that is really the limit. But I would say for that point, investors, at least what I experience, is that there is definitely also an understanding that in CMT, there's really a high unmet medical need that's not addressed at the moment. So investors understand that. So they see CMT to be an area where it makes a lot of sense to invest in new drugs that's going towards CMT. In terms of how big is the pipeline, I would actually say that it's smallish at the moment still. We saw the nice slide before where we saw the attrition rate. If you take that attrition rate and you combine it with the many different subtypes of CMT, I think, and I hope we will see more. I hope that pipeline is going to expand a lot more. I think it needs to before we get to where we want to be. So I think that's just that's my take on it.
Oh, great. Thank you so much, Thomas. I think just so, some of the patients understand, to do a single animal study over the period of time for looking at some of these neurological disorders, it can cost somewhere between $100,000 to $200,000. And to a biotech company, when you have to do this with multiple entities over multiple years, I mean, that's, those types of costs add up and make it challenging. But there are so many costs once you start moving into the clinic and you start recruiting patients. And so certainly significant funding is necessary. Artie, there are a number of patients here today, thankfully, both here in person and online at this meeting. As a drug developer, what would you like to ask these patients if you had the chance to kind of make a request of them today?
I'm going to add some of the answer to your other question.
Sure, you go right ahead.
But maybe I'll start. Yeah, I mean, I consider myself a drug hunter. My mom is really proud of that. But in all seriousness, I think for the patients, we're getting close. We're getting really close to entering clinical trials. DTX, we're less than 12 months away. Other people are coming right behind us. And so you've done an amazing job. And it's really rare in drug development to have 10 years or more of groups like the Inherited Neuropathies Consortium and the patient contributions where you have target engagement assays so we can measure for CMT1A, PMP22 suppression. There's biomarkers that have been identified because you all have been going to your doctor every year and allowing them to do research. There's scoring systems, and all these things are, if you're a drug company, you have to have ways to show at different stages of development that your drug can work. And we're looking for tiny signals, hints, clues, whatever. So I think my message would be, we're like 12 months away. We're cramming now. Every KOL in this room we're picking their brains on how to run these trials faster. Is there anything we can do in this 12-month period to learn as much as we can? So my ask is, keep enrolling in these natural history studies, the biomarker studies, even the FARNEX trial, we're going to learn so much from at DTX to make sure we're successful. So that's one ask. And then I don't know if this is selfish or not, but I think they're all, but you guys always are telling me stories. I've probably collected 20 or 25 stories. And so where they're helpful is inspiring our team to keep coming on the weekends, to come in at night, to keep working on this project. And so I've heard a number of stories this year. So keep telling us the stories, keep sharing the stories. It helps us think of ways to help you. Now, I want to come back to the last question, if I'm allowed.
Go ahead.
I think I want to talk about pharma, and I want to talk a little bit about the institutional investors. So this is $1 billion opportunity, CMT1A. There's 75,000 patients in the US, 150,000. You don't even have to charge a rare disease price to get to $1 billion. So I think the short answer to is pharma interested or institutional investors interested is absolutely. They're really interested. They're really excited about CMT1A. It just takes this kind of activation energy that's in this room to show them, having the clinical path sort of paved for them. And it'll be the biotechs that pave the way, and they'll come and buy all of us. And, I guess my last point is for SMA, there's a small molecule. So right there, there's an ASO. So right there, and there's a virus approved today. And all of them are doing just fine. And the patients, that are taking those are doing really well. So that's my way of saying that there's not enough competition yet because we don't have 3 things on the market yet. So
I'll add to that in terms of the pharma. Someone asked me today, how did I get involved with the CMTRF, and when did I get involved? And actually, my answer was it was because of Pat Livney and Susan Rudiger, and because of they approached me when I was at Sanofi. And so at Sanofi, we worked with Pat and Susan to really drive a program, and there was interest there. We didn't really have a modality that we could advance, but I'm sure once we get to this point, there's a lot of interest once you hit clinical trials and you have clinical trial success. So we'll see. I think you're right. We will see that sort of increase in terms of the interest. So I'm going to open it up to all of you now. So please just raise your hand if you want to answer the question. And we've probably talked a little bit about this, but I'll ask it again a different way. What does make CMT an attractive disease for biotech and pharma companies? Why would they be interested in this area? Anybody want to take that one? Okay.
I don't want to take.
Go ahead.
I pitch this story every day, so I
Sure.
feel pretty passionate about it. I think, one, we understand, at least for CMT1A and some of the other CMTs, we understand the disease-causing lesion. So we know the genetics. And so if you can get to the genetic lesion, that is always very attractive. You understand what's causing the disease. I would say that the 4 things I alluded to earlier on, right? The last 10 years of research has identified ways to measure suppression of the gene through skin biopsies. There's, I think, 3 circulating biomarkers identified. There's the structural biomarker, the CAF-FAT MRI. There's the nerve conduction velocity action potential test that I know everyone loves, but is really helpful and will be really helpful to guide. And then, of course, there's scoring systems that, there's many of them. But I think we're all trying to figure out which is the best one for the kinds of trials that we're all going to run. But that's attractive. There's checkpoints at every point in drug development for this program and natural history data that we can lean on. So we're not flying blindly when we get into development. And I can tell you that the first 3 indications DTX looked at, we were going to be flying in the dark, and we had to abandon them because we didn't know, we couldn't convincingly tell people that we would get an answer for them.
Anybody else?
Well, I can just say that it's, the usual things that sort of one would look at for evaluating whether this is an area that's of interest for a pharma company, maybe, is there an unmet medical need? Is there any treatments that really is kind of crowding the whole space? And in both cases, in CMT, you can say there's a very clear unmet medical need here. And is there a lot of drugs out there already approved for CMT? And the answer, of course, is no. So this is something where there is a very clear requirement or a need for new treatments, and there is space for it. So so I think that's just very important.
And just a little follow-up too. I think that, say, even one or two drugs wind up being approved, even in the CMT1A space, you can see that not all patients are the same. And so simply having one and hoping that one drug will cover everyone in CMT1A is probably poorly guided. I mean, I think what you want is coverage. You want to have multiple shots on goal. And why are they different? You know, I think we're still a long ways away from understanding stuff. And so let's not put, you know, the cart before the horse. Let's get everybody to the finish line and then start sorting things out, whether it's multiple ASOs, multiple small molecules, gene therapies. There's so many modalities that when you have a clearly identified genetic target, man, that's the best possible thing you can have. So I think it's really exciting to be able to say, let's get multiple things over the finish line and then worry about that later, because I agree. I think the more competition, the better. And do you really want to be standing all by yourself in a field because no one smells any opportunity? You want to be in a crowded space because there's an opportunity there.
There's a lot of synergies to be had too, from all the work at the different companies, biotech, startup, pharma. At least I've overseen rare genetic disease portfolios at multiple companies now. And our perspective has always been, we look in the future that there will be combinations therapies. And that's probably where we'll end up with a lot of these rare diseases, is there will be combinations to treat the patients and give them the maximal benefit. All right, so I'm gonna end one last question here before we open it up to the audience and an online audience. You've been at this meeting today. How do you see how private philanthropy and nonprofit patient organizations help your company work or can help your company?
Well, I can start. So, I mentioned that the work that we're doing initiated from an academic lab. So the very first thing is, and I also mentioned actually that I tried to get funding for CMT1A gene therapy project 15 years ago, and again thereafter, and it didn't pan out. So one of the very first things that comes to mind is, CMTRF stepped in and funded this project that just a few years ago that Cleopas and I were developing, and really kickstarted the project. When we struggle to get funding elsewhere. So, that to me, that's the very first thing that comes to mind. That the foundations sort of fill that, help us step through the breach and develop something new. And we have to generate some data to generate proof of concept, to demonstrate that what we're trying to accomplish is actually feasible and going to work before pharma or even, at earlier step biotech gets involved. And so, Armatus Bio, this is a startup company that was founded by a bunch of people who have experience, serial entrepreneurs, experience developing companies. They saw the promise in what we were trying to do and licensed the work, and they're helping us translate it. So without having the initial funding for this project, and it was a lot of money, but it wasn't pharma money. It was I don't even think it was a couple $100,000. It has put us now in a position where we are able to maybe translate this soon.
So there's a valley of death earlier before you get to the other valley of death.
That's right.
Before you even can kind of translate into a specific indication, and the nonprofits helping to bridge that.
Absolutely.
Yeah, great. Anybody else? Any comments? Yes, Sylvain.
Yeah, so definitely working with patient advocacy group is really important. In my previous company that I founded some years ago, we were funded by Minister of Defense in UK, but also the Wellcome Trust and also Action on Hearing Loss, because we are developing drugs for hearing disorders and tinnitus. And without their money, without this non-dilutive funding, which is still, I think, important to generate the first set of data that will convince investors, that will convince pharma companies to make I would say, collaboration. But the patient voice is very important for us. This is how we start most of the time, is what it's needed and what we can generate in terms of data. And I've been to work with this patient advocacy group in different fields in the past. And the most, I would say, to be proud of that is that when we are successful and with a return, then this money that are giving back to the patient advocacy group to fund new projects. And this is how it starts. And I think pharma companies look like as a strength, because you are already engaged with this patient advocacy group to understand the disease itself. So it's really important. Yeah.
Absolutely. And we hope that happens as we continue to fund projects. So we're going to shift gears to our third phase of this panel. And we're going to go to some of the online questions that you have, Rob.
Yeah, I'll ask two questions first. And this is to everyone. First of all, super excited about all the work that you are presenting. It looks like therapies will be hitting to the market. But two things. Have you seen during this conference any subtypes of CMT where you think, my technology is applicable to this, and that you may broaden other subtypes into your research? And two, do you think that at a certain stage, going to clinical trials, that not only would you be able to prevent progression, but maybe reverse clinical symptoms, or is that a bit too much to ask?
Anyone, or I'll have to pick somebody.
I can start. Where we started the project with H6 inhibitor, it was a prevention. So we were starting to dose the compound in a model where they are already, we say, the symptom that's not there is progressing at the same time. So as a prevention. We have now the data that can also be interesting to look at the curative treatment. And so this was the initial set of data that Ludo Van den Bos, our scientific founder, found in the CMT 2D. So he worked on the HSBB1, GARS, multifusin 2, and he found that, in fact, is a curative treatment. It regenerates the neurons, is induced reinnervation, remyelination, and all start with there's a potential cure somewhere down the line. And what we try to replicate is to do in all subtypes to do the same. And the CMT1A will be, I think, the first project where we do a long treatment, curative treatment, where the symptomatic mice are already there, and we try to reverse everything. So definitely there's a hope that we can, what we found in CMT2 can be applied to CMT1A.
Oh, that's fantastic. Oh, go ahead, Scott.
Yeah, so hopefully the SWORD trial works great. And I mean, I think it's super promising. Like I said, I'm sort of new to the field. We've published a couple papers with collaborators here over the last couple of years, but I'd still consider myself a newbie. So the SWORD data I hadn't seen in depth. I think it's fascinating. So congratulations on that study. And hopefully, like I said, the phase 3 works and we don't need it. But I was thinking, well, geez, I wonder if we could do gene therapy. Yeah. Sure. And I'm sure, you've thought that as well. So as one of the talks was going, I was on my phone looking up, are there mouse models? How big is the cDNA? Does it fit into a virus? That sort of thing. And then the other thing I'll mention about this meeting is, I mentioned we're doing a CMT2D project, and we've hit a few bumps in the road on trying to get this GARS gene re-expressed the way we want it to, at the levels we want it to. So, we've got an expert in the back standing there, Dr. Antonellis, who's given me a few pointers on maybe how I can improve the design of our vector. So, there are lots of ideas coming to mind here just being at this meeting.
Oh, it's fantastic. And thank you all for being here today too. I think your physical presence here and the discussion is just really hopeful, brings a lot of hope to a lot of patients. Rob, do we have any other questions online?
One specifically to Scott, and it's just about targeting Schwann cells. And do you feel that it's a difficulty in specifically just targeting them with AAV, but
Yeah, is it difficult to target Schwann cells? Yes, yeah. So one of the big questions we get, going back to this notion of, is pharma interested? They are. We talk to pharma a lot, and but one of the questions that always comes up, and it's a legitimate question, is everything we've done so far has been in a mouse, and the nerve of a mouse is about this long. So if we're doing an intrathecal injection in a mouse, the virus only has to travel so far. Will that virus travel down the nerve in a human being? We don't know the answer to that question. So that's a major push. We need to prove that. And so I was really encouraged. I actually asked the question during the scientific session yesterday. Rachel Bailey gave a really nice talk and alluded to, didn't present any data, but alluded to a patient who had died, who had been enrolled in the giant axonal neuropathy gene therapy trial. Didn't die from the therapy, died from something else, but they were able to get autopsy tissue from this individual. And they looked and they said, she had mentioned that they had found the virus down at the distal part of the nerve in the human being after an intrathecal delivery. So that was encouraging. We haven't seen the data. I asked her the question. She said, you wait for the publication, so, which we will. But, so we need, one of the things we're trying to do is, number one, I mentioned earlier, develop better vectors that can hit more Schwann cells. But number two, just ask that basic question. If we do the delivery we intend to do, can we hit the Schwann cells all the way down at the end of the nerve? We just don't know yet. Okay.
Any other questions online, Rob? Okay, so now what we're going to do is we're going to turn it over to the audience here, and we have runners with microphones, and we also have a microphone at the center. And we have these brilliant scientists and executives here that are willing to answer any question that you have about, CMT drug development, or anything that they're working on. So we have any questions? We have one right here. Okay.
Thank y'all for being here and educating us. Can you tell us exactly what Schwann cells are?
Okay. Well, I would, to simplify it, I would say that, there are two parts of the nerve. So there's the nerve part, the neuron, and then there's the Schwann cell that sort of wraps around it. And there's something called a myelin sheath that protects the nerve.
Drawing a doodle of it.
Oh,
This is the
okay.
myelin sheath right here.
And I've
So
studied,
some CMT mutations affect genes that are involved in this, Schwann cell's ability to make myelin and protect the nerve. And some genes are involved in, different aspects of the biology of the neuron itself. So, the viruses we're using can target both the axon and the Schwann cell.
thank you.
We have a question back there in the back of the room.
Thank you. Just comparing mouse models to human models. I know when I was diagnosed with Charcot-Marie tooth, I had an MRI done. It showed a lot of fatty infiltration in my muscles and things of that nature. Is that seen in mouse or rat models in terms of the neuromuscular degeneration or degenerated muscle?
Me again?
There's definitely muscle atrophy. I was actually wondering myself, because we used to use these MRIs and obesity research to measure fat in the tissue, but at least to my knowledge, I'm not aware of any publications that show fat content of muscle. But when I get back to the lab, I'm going to see if we can find one of those machines or do some sectioning. And I guess with respect to the translatability of the mouse model, we think it faithfully recapitulates most aspects of the disease in terms of nerve conduction, action potentials, grip strength, balance, et cetera, and the degree of demyelination. The thing that it probably doesn't, at least the mouse model we use, the C3 mouse, it probably doesn't capture the axonal loss that happens potentially later in the disease, and perhaps some of the sensory components, to my knowledge. But
And maybe I'll add just one more comment. So I think also part of your question is relates to how much can we trust what we learn in the mice, and how likely is that then that they will be having a benefit then for the patients in the end. So I think what we heard this morning, Melissa was speaking about the observational study, where you also have the clinical trials, and the observational studies are exactly the type of studies where you try to get an idea about a particular element of the disease. And ideally, you would be able to do that also in the mice, so you can have a confirmation that some of these things do translate. Specifically, in our case, I can say that we are measuring exactly the ability of the motor nerve to transmit the electrical signals into muscle. This is a very well-established technique that you can apply in mice as well as in man. So we have done it in mice, we've done it in man, and this is because we can now see that kind of deficit both in mice and in man. We can correct it in mice. That gives us the comfort to say, okay, it looks like we have that kind of deficit in man and in patients, therefore we go ahead. So those observational studies are key for addressing the kind of translatability that I think is very important.
And that refers to CMAP, restoration of CMAP is that
Yeah,
right?
exactly. So you basically repetitively activate a motor neuron. You look at whether the CMAP is translated every time, and sometimes they don't. And if they are not transmitted, that means that somehow there is a dysfunction between nerve to the muscle communication. And we can see that both in the animals and in patients. And importantly, in the animals, we can correct that. So this gives us kind of a composite data set to say, well, there's reason to believe that we should be moving forward here in CMT with our type of mechanism. But I mean, this is just one of the mechanisms, more from a kind of, principle idea that we can sometimes get informed from these clinical observational data.
And I think too, that when you see that inserting the human gene for PMP22, that gives you a lot of targets that are important, humanized essentially. And then like what was mentioned is you see a lot of the disease characteristics recapitulated and then comma, but it's a model, but so it doesn't have to be perfect. And I think that the understanding that it is a model and will not be 100% CMT1A is perfectly acceptable. But that the conclusion of clinical trials will really start to like lay over the models and what they represent and how important particular characteristics are for those models that were used to get to the clinical trials. So I think everything that's learned here going forward will be invaluable for the folks who are behind you picking a model and trying to overlay it to a clinical trial.
And I'll just say one thing about that. When you think about the nerve, it's much like an electrical wire that is actually, sort of running, a motor. In this case, the motor is the muscle. And as that electrical wire sort of can't send those electrical impulses down to the motor, what actually happens is in the case of both mice and also humans is that muscle will atrophy because that loss of that neuromuscular junction, that connection, the muscle will actually shrink. And you see that in the CMT mice, you see that in patients as well. I mean, we all know that with CMT. And, and so we do look at changes in the muscle diameter change in the clinic. And that will probably actually be one of the biomarkers that's used to actually measure clinical efficacy of molecules as they go forward.
Hey,
Go
sorry,
ahead,
Paul.
Pat. Yeah.
I got a quick question. First and foremost, you guys are phenomenal, and I'm very proud to have you guys on stage on behalf of the CMTRF. I just got a really silly question. 5 years ago, what were you guys doing?
Why don't we start from left to right and go down? So, Chris?
I basically did not have a company, and I exclusively did spinal muscular atrophy research. So now we've expanded into other diseases, and CMT1A and 2E are an important focus.
So yeah, I was making drugs. I'm a medicinal chemist by background, so training. So I make drugs for the past 20 years. So I know how to make it drugs for the CNS disorders, and now I'm making it for the PNS and all these CMT fields. So yeah. we'll continue to do that for the next generation. 5 years ago, so NMD comes out of my own PhD back in the day. So it's kind of really of a story of an academic idea that came out of a very basic scientific question that led now, and now we're in phase 2. So it's kind of a nice story in some respects. So 5 years ago, I was chasing funding, I think. That's pretty much what I was doing. So I was looking for raising the first seed round. At that stage, we have formed a company, but we were in our sort of second seed funding raise. That's what I was doing.
I was sitting in my room by myself, thinking about how I was going to raise money for DTX, and we started raising it $5,000 at a time. And about 5 years and a month, or maybe backwards, we moved into like 100 square foot lab, one bench. So.
Well, that question gives me an opportunity to talk about my backstory a little bit now. So thanks for teeing that up. 5 years ago, I wasn't working on CMT. I was just entering the field, and I'll be quick about it because I think I droned on about this last night at a different event. So I primarily work on muscular dystrophy, and I started doing that 25 years ago in my PhD, and continue to work on that now as a professor. 5 years ago, I was associate professor, not a full professor, and I alluded to a couple times that I've been flirting with the CMT field for a lot of years. About 5 years ago, I got an email out of the blue from a gentleman who he's a neurosurgeon at the University of Texas, and he had a granddaughter who had a form of GARS CMT2D that was actually originally diagnosed as SMA. It was such a severe case, and he said that he heard I was working on CMT, and I wasn't. But the reason he got that idea in his head was because I had applied for this grant when I was a postdoc 15 years before that didn't get funded. And I moved on to my own job and started working on muscular dystrophy. But I don't really know how he found my name, but I suspect somebody who reviewed that grant remembered the idea and said, oh yeah, this guy might be doing something in this vein. Long story short, probably everybody here who's been in a position to develop drugs and publish papers has been contacted by somebody saying, hey, can you create this therapy for my own family member, right? And usually, probably most of us get those and politely decline because it's not a focus of ours. But something about this grandfather kind of tugged at my heart, and I thought, well, maybe I should actually look up the disease that he's wanting me to treat before I turn him down. And I did, and it was CMT2D, and I looked up some papers and found a mouse model or several mouse models that had been created or analyzed by this guy, Rob Burgess, who many of you probably know is at Jackson Labs, leader in the field. Never met Rob before. And something just kind of compelled me to say, hey, let me email this guy, Rob Burgess, and see if he wants to work on this project together. I can do the gene therapy part. He can do the mouse model part. And that's it. I emailed Rob. Next thing I know, we're having a phone call and trying to get funding. We got funding from MDA. We got funding from NIH. And we have this therapy now that's Armatus is licensed. Because of that, so that's how I got back in the CMT field, by working on that project inspired by this grandfather. And then fast forward like two years later, I'm at another meeting, a CMT meeting, and I run into Cleopa there, met him for the first time, and we started talking about CMT1A. And because of those connections, connecting those dots, now I'm working on CMT1A with Cleopas. So, you don't, I guess, never give up on what you think you want to do for your career. And here we are 15 years later. So 5 years ago, I was not working on CMT. I am now. Two different things. Yeah.
So just to kind of wrap here, I think you've heard about all the new therapies that are being developed. And we're lucky to have these executives here with us. Their hard work and millions of dollars that are being invested in developing therapies for CMT. It's not rocket science. It's actually harder than rocket science, right? I think that's true. I just want to say, we are thankful for your efforts. Both, I want to, on behalf of the patients and the CMTRF, as Pat said, we thank you for your continued efforts and we wish you the best of luck as you go forward. So I'm going to turn this over back to Cleary Simpson now. Thank you all for your being panelists, and we'll transition to the next schedule.